Hello, everyone, and welcome to Beyond Opiates, New Horizons in Pain Research, a special presentation from RCI Science and the Gairdner Foundation. My name is Carrie Boyce, and I'm the Executive Director of the Royal Canadian Institute for Science, here to welcome you today and get a few quick housekeeping notes out of the way. So today's format will see a short presentation from Gairdner laureate Dr. David Julius sharing a brief look at current and future research goals and challenges associated with achieving better pain control. Then Dr. Janet Rosson, president of the Gairdner Foundation, will moderate a discussion incorporating your questions. So with that, please don't forget to submit your questions in the chat box or the comment section, depending on what platform you're joining us from. You'll likely need to be logged in. So if you don't have an account on either YouTube or Facebook, you can also submit your questions via Twitter by tagging us at RCI Science, or you can email in your questions to information@rciscience.ca. Don't worry if you don't remember all that. A message will be on screen during the presentation to remind you. And also just a quick note to please remember to be polite and respectful in the comments or the chat box. Moderators will be monitoring the conversation and taking action as appropriate. Now, if we're lucky, we'll also get to play a quick round of some questionable news, again, where we take a lighthearted poke at some science-related headlines. In case you missed it, earlier today, we dropped our very first episode of Some Questionable News, a web series that does just that. You can find that on our YouTube and Instagram channels at RCI Science. But that's plenty from me. I told you I wanted to be quick. So without further ado, it's my pleasure to hand over to the president of the Gairdner Foundation, Dr. Janet Rosson.
Thank you, Carrie. It's great to be here on behalf of the Gairdner Foundation. I'm Janet Rosson, and I have the privilege of having a great job. I'm the president and scientific director of the Gairdner Foundation. Now, the Gairdner Foundation has been around in Canada since 1957, founded by Jim Gairdner, a Toronto philanthropist, who wanted to recognize those people whose research has really had an impact on human health. And over the years, since 1957, we have been handing out international awards for biomedical research that have really encompassed so many areas, from fundamental biology to infectious disease to vaccines to cancer treatments. And we've given out over 350 awards, and 95 of the people who've received International Gairdner Awards have gone on to receive the Nobel Prize. This is a really highly respected international award, and we have fantastic laureates who have received our award, including, of course, this evening's speaker, Dr. David Julius. So David Julius is professor at UCSF, and you're going to hear all about his research. He received the Gairdner International Award in 2017 for his work on really trying to understand how we receive signals from the outside world, particularly heat and cold and noxious signals, and discovered receptors, pain receptors, that pick up these signals. And what he's going to tell you about tonight is about those pathways, and also, of course, how understanding how these things work can give you new ways of developing pain control. Now, David received the Gairdner Award in 2017, and it seems that he's been on a roll ever since. So he's receiving lots of other awards, so we have great hopes here. He received, let me get this straight, the HFSP Nakasone Award in same year, I think, 2020. Oh, I may have got that one wrong. David can correct me. The Breakthrough Prize in 2020, the Kavli Prize, and the BBFA Foundation Frontiers of Knowledge Award in quick succession over the last few years. And all for this amazing fundamental science that has great potential application for human health. So I'm not going to take any more time. I'm going to hand you over to David and let him tell you about his research.
Thank you, Janet, and thank you, Carrie, for inviting me to speak to everybody today and tell them something about what I'm thinking about this area, a little bit about our own work, but mostly about this little presentation, brief presentation is mostly to, I think, address what some of the questions are in kind of modern day pain research, and to discuss some areas that I think are sort of, either being pursued now or up and coming, and some really important avenues of research that people in the pain field, not just myself, are headed towards. So let me share my screen with you. Whoops. Here we go. Okay. All right. So, the first thing that I wanted to address, why do we experience pain? We all think of pain as being something of a negative value, but we really, the pain system is really so important for maintaining our health, because it's the main warning and protective system that tells us when we've injured ourselves or when we're about to injure ourselves, and then to initiate what we call, appropriate protective reflexes. So, for example, you learn that if you touch something hot, you need to either avoid touching it, or to pull your hand away quickly. And we know through genetics, namely people who have mutations that render their ability to sense pain null, that is, their pain system is not operating, that we need this system for our survival. So in those rare individuals who cannot sense pain, they are at great risk of injuring themselves, and even at great risk of death. So I often say that among our sensory systems, including things like vision, taste, smell, etc., that our ability to experience pain is maybe the one that's actually most important for our survival and our health and well-being. The problem is that while acute pain is very protective and essential for our bodily health, we all know that this system can become persistent and debilitating. That is, that acute pain can become chronic. And when it becomes chronic, it really outlives its usefulness as a protective role, and really upends our lives. So a big goal in this area that we work in is to understand how this transition from acute to persistent pain occurs. What are the underlying physiological and molecular mechanisms? And the ultimate goal is to either reverse this with injury, or to prevent it from happening altogether. So the challenges are to really, from a basic science perspective, is to really better define different pain syndromes. How are different chronic pain syndromes, how are they different, and what are the underlying molecular and cellular mechanisms that account for this? So what I mean by this really is that when we think about different types of chronic pain syndromes in particular, we use the word pain or chronic pain as sort of one entity, but it's much like using the term cancer. We know now that there are many different types of cancers, and that there are different molecular and cellular mechanisms that underlie distinct types of cancer, such as bladder cancer or bone cancer or brain cancer. Well, the same thing's really true in the pain system, that while we use this single phrase, there's lots of different types of pain. There's musculoskeletal pain, there's lower back pain, there's migraine pain. There's pain that's associated with inflammation, such as osteoarthritic pain. And then there's pain that results from viral infections, such as shingles and herpes zoster. So each of these pains almost certainly involves not only different etiologies, but different molecular underpinnings. And we really can't begin to think about therapies in a rational way unless we understand how different pain syndromes differ, because having distinct mechanisms means that there will probably be different pharmacological remedies to treating these different types of pain syndromes. Maybe not all of them, but at least some will differ from others. So one of the goals is to really understand the different mechanisms, cellular, molecular, physiologic, that underlie different types of pain syndromes, so that we can really understand what the basis for these different maladies are. Now, the pain system's complex, like most other neural systems, and it involves neurons both at the periphery, namely, for example, sensory nerve fibers that would innervate a finger or any peripheral site, could be your lips or your nose or your arm, etc., and even visceral and vascular organs such as your bladder or gastrointestinal tract. And these neurons send their connections into the central nervous system in the spinal cord, where then they engage in communication with other so-called ascending neurons that convey this information from the periphery to the brain, so that cognitively you understand that when something noxious has happened here, that you perceive this as a painful situation. And many of the questions that in the field we're still engaged in are in some ways complex, but also simple questions. And that is to ask, how many different types of sensory nerve fibers are there? What are they sensitive to? Are there certain ones that are sensitive to hot or cold or pressure or chemical agents specifically? What kind of target tissues do they innervate? That is, do these different neurons receive messages in the finger versus the bladder versus the lips or tongue or face or other places, and what are the different contributions of these different sensory nerve fiber subsets to different types of persistent pain, like, for example, migraine pain versus osteoarthritic pain. Similarly, as we go further into the nervous system, we want to know, for example, in the spinal cord, how many different types of nerve fibers are there. The spinal cord is incredibly complex and contains a lot of different neuronal cell types. And really, we're just beginning to understand what those different cell types, what roles they play in contributing to the conduction of pain signals from the periphery to the brain. What are their sensory partners? Who do they talk to from the periphery? How do they connect to the brain? And again, how do these different subtypes contribute to different types of persistent pain? And while injury in the periphery can lead to chronic pain syndromes, injury within the central nervous system, such as a thalamic stroke or other sorts of catastrophes like that, can also lead to terrible chronic persistent pain syndrome. So we want to understand this neural network. We want to understand the molecules that make these different sensory neuron and spinal neuron subtypes distinct. And this will give us clues as to how we can target them. Now, we all know that there are drugs that everybody takes to control pain. But the problem is that the repertoire of drugs that we take commonly are limited. So, most of what we take are every day at least are found within the categories of so-called opiate drugs, things like morphine and related molecules, or the so-called NSAIDs, the non-steroidal anti-inflammatories, or acetaminophen. These are things like aspirin, other inhibitors of enzymes called cyclooxygenases. The problem with these drugs is, one, they don't work on all types of pain. So, for example, if you have bone cancer pain, which is a very intense sort of pain, opiates and NSAIDs are only partly effective. And this is true for a variety of different pain syndromes, migraine pain, for example. The other main problem is that these existing analgesic drugs have a lot of side effects. So, as we all know, opiates have effects that range from respiratory depression, that is, they stop breathing, which is why people die from opiate overdose, to constipation, to behavioral syndromes such as tolerance, addiction, etc. And we all know this from the increase in opiate addiction and the opioid epidemic that's been affecting much of North America and other parts of the world. Non-steroidal anti-inflammatories like acetaminophen or ibuprofen have toxicity at internal organs like liver and kidney, for example. So while these drugs have been really fantastic for treating a variety of different pains, they have their limitations in terms of their efficacy in certain types of pain and their side effect profiles. So one of the goals, for example, of the work in my lab and many other labs is to focus on, for example, these sensory nerve endings and to ask what types of molecules they express that contribute to the detection of painful events, and that might be involved and that might alter their activity in the context of injuries or inflammation that then lead to persistent pain syndromes. And one of the reasons to focus on the periphery is that the further out you are from the central nervous system, perhaps or other organs, there's more of a chance that you will have a specific mechanism of drug action. So as Janet mentioned, we have focused on identifying molecules, for example, that are activated by temperature, like cold or heat, as well as chemical irritants, and trying to understand how they contribute to the activation of these nerve fibers from a very basic curiosity-driven perspective. But we've also found that many of these targets are altered in the context of injury, particularly inflammation, such as arthritis, that then leads to persistent pain that will eventually manifest itself as what we call maladaptive or chronic pain. So we'd like to understand how these molecules work and how we can take advantage of this information, not necessarily we, but people who are interested in drug design, to develop drugs that can be used to combat a variety of pain syndromes, such as osteoarthritic knee pain, migraine pain, or other types of pain that might involve tissue inflammation. So we want to identify these relevant so-called receptors on these nerve endings and identify drugs that act appropriately. And in one example, at least, of the work that we're doing, we're using molecular and structural methods to identify the molecules that we've looked at. Here's a receptor for a compound called capsaicin from chili peppers that induces pain, that has identified this receptor that's also activated by heat. Here's another molecule that we've worked on that's activated by pungent agents in wasabi and other types of mustard plants. And this too is a receptor on these sensory nerve endings that's involved in detecting chemical irritants in our environment. And by obtaining the atomic structures of these molecules, we're able to see where drugs bind in these little pockets, and to better understand how these can serve as so-called druggable sites for inhibiting their activity under circumstances where they become hyperactivated and lead to persistent pain syndromes. Now, other approaches that many labs around the world are taking are, for example, to exploit what we call computational chemistry, as well as to continue to exploit natural product chemistry as we have done to develop analgesics. And just as an example, one idea would be to take a receptor that we know and love, let's say an opiate receptor, where we know that activation leads to analgesia, but there are also side effect profiles associated with binding of this drug, and to ask whether molecules can be identified that activate these same receptors but favor the pathways, the cellular signaling pathways that lead to analgesia, and spare the pathways that lead to side effects such as respiratory depression. This is an area that people are very excited about, but it's really still in its infancy, but involves the identification of new drugs that can somehow activate these sites in ways that give them specific properties to signal through specific pathways. And part of this is to really not only design drugs in the lab, but to continue to mine our natural environment for natural products through folk medicine and other forms of discovery that we know through experience can be used as analgesics, and to better understand the mechanisms that these plant or animal-derived toxins and other compounds use to affect the nervous system. And this is the kind of work that my lab's involved in. And it's really fascinating work because it sort of sits at the interface of, anthropology, chemistry, and physiology. And the other thing I would say is that this is sort of a pitch for preserving our planet and preserving all of the wonderful natural products that nature has given us that we can exploit for these kinds of drug discovery efforts. The other approach is really an engineering approach, bioengineering in different ways. And I've just pulled out a couple of examples from efforts that some of my colleagues here and other places are using. And that is, for example, to use stem cells, so-called iPSC-derived stem cells, whereby cells that may have been injured, for example, in the spinal cord, that control pain signals can be reestablished by injecting iPS cells into injured areas of the spinal cord to return, for example, inhibitory input to diminish pain signals and to restore the natural circuitry of the spinal cord that may have been disrupted, for example, in spinal injuries that lead to chronic pain syndromes. Another one is to implant electrical devices, and there's a lot of work in this area that's really improving over time, to stimulate pathways that have lost input from the periphery, or to modulate those pathways to, again, modulate the control of pain, either through what we call descending inhibition of the pain pathway, or other ways that we can use to control chronic pain and reestablish normal circuitry and signaling in the nervous system. But in order to do this, as I said, we really need to understand the basic neurocircuitry of the pain pathways so that we can really provide some evidence-based support for such approaches and take them out of the realm of just being sort of, randomly observational and proceeding with luck, and really understand what the molecular and cellular basis is that will help us improve on these kinds of strategies. And then the last thing I want to mention is that is our ability, and what can we do to improve our ability to measure pain objectively. So one of the big problems in pain research and in the clinic is that pain, of course, is a very subjective and variable phenomena. It can also exist or persist without signs of physical injury. So even if after a physical injury has resolved, people can still be left with pain and discomfort. For example, in things like irritable bowel syndrome, there's no longer evidence of inflammation to the gut, but people are still left with some very serious visceral pain syndromes. And as a result, and also the diagnosis, of course, in the clinic is largely declarative. You have to tell the doctor what you're feeling, and sometimes it can be difficult to quantify or localize. And this has really raised the challenge of developing tools that can be used to assess pain more objectively. And there's a big effort now to identify what we refer to as biomarkers or molecular probes to measure pain. This is really an area that it's at its infancy, and we're still asking questions about what are appropriate biomarkers to measure pain, both acute and chronic pain. And then the other approach is to use imaging methods that we can use to more objectively assess activation of neural circuits and neural pathways that are involved in pain, to provide signatures for specific types of chronic pain syndromes, and to objectively measure pain for clinical trials. So clinical trials are really beset with the problem that there's a very large placebo effect. And so sometimes it's difficult to assess the efficacy of new pain medications. The placebo effect itself is a very fascinating thing and can potentially be used to harness the body's own mechanisms to fight chronic pain. But we need to understand when we're measuring a placebo effect and when we're really measuring a true drug-evoked effect. And objective measures would also be, incredibly useful for measuring treatment, for treatment assessment, not only in clinical trials, but in the clinic. These sorts of efforts are also being initiated with the long-term idea, that if you can assess pain more objectively through imaging techniques, you can also couple these with biofeedback devices that can be used to control pain in patients. So I hope I've convinced you in this little talk that really it's that basic curiosity-based science in terms of understanding basic mechanisms and properties of the pain pathway is really fundamental to achieving better pain control, because we need to understand what the cellular pathways and molecules are that we must target in order to treat different types of pain syndromes, and that we need to better define different types of pain syndromes to understand how they differ and how they can be targeted more effectively and specifically. Thank you very much. Happy to take any questions.
Thank you, David. I think we're going to go to some questionable science first, are we not?
Oh, yes.
We sure are.
This is the part that really makes me nervous. Okay.
This is the part that it shouldn't make you nervous. This is just a little bit of lighthearted fun. So if you're new to this is a game we like to play, putting some of our expert speakers on the spot. We will display a headline on screen, and you and our viewers at home will vote on whether you think it is a real headline or something ridiculous that we made up to try to catch you out. So you can play along at home, you can type along using the live chat or the comments box. But without further ado, we'll get on to the first headline. Mice may catch each other's pain and pain relief.
Mm-hmm. Do
you think that is a true, real headline that exists in the world, or something we made up? Not whether it's factually accurate, but just whether as a clickbaity headline, it might exist.
That's a low bar, but I think that this was probably about empathy, and I would say yes, it's a real headline.
What about you, Janet?
I probably would agree.
All right, and our audience seems to be voting true as well. We can confirm that it is real. Yeah, this was in Science News. It wasn't actually. It actually was a pretty good article, I have to say. So you can find that if you'd like to learn more. This next one I really enjoy. Does your pain feel different in English and Spanish? What do we think? I'm going to have to start putting a timer on this.
I think it's made up, but it's certainly true that people make different sounds when they're in pain in different languages.
Mm-hmm.
But I don't know whether that means the pain feels different. So I think this is made up.
Yeah, I agree. I think people describe pain in different ways, but I think they feel it in the same way.
It's real. If you can believe it.
But it's real.
It's real. So this was actually a study out of the University of Miami suggesting that language of bilingual speakers can affect their pain depending on their cultural associations that they have to each language. So individuals reporting more intense pain and showing larger physiological responses with respect to heart rate and sort of sweaty palms and whatnot when they spoke the language of their stronger cultural identity.
Oh, I see. Okay.
Group size of about 80, not definitive, but certainly really, really interesting kind of early conversations. Okay.
Hard to know if they were really feeling it differently or just more comfortable expressing it in their native language.
Yes. So they had subjective feedback as well as sort of a few physiological markers.
This is where an objective assessment would be very beneficial.
Indeed.
Exactly. I was going to say that comes to your point, David, about how difficult it is to measure pain objectively.
So this next one, report stubbing your toes 7 times more painful than childbirth. Those who have birthed a child may have something to say about this, but do we think this is a real headline or something made up?
Made up. Certainly not true.
I'm unqualified to answer this question. I'll go with Janet.
All right. It is indeed fake. Well done. Mothers out there declaring it couldn't possibly be true. All right. One more just for luck. Why redheads feel less pain, according to scientists.
Oh, yes. This is a Canadian study, I believe, from Jeff Mogul, maybe. And I think it has something to do with linkage to a peptide called CGRP or some such transmitter. So I think it has to do with the genetic linkage to a signaling molecule that's involved in pain sensation.
Janet, I assume you're sticking with that? Yeah. It is indeed real. This was in the New York Post. And that is a more accurate summarization than what was found in the New York Post article.
Like the secondary headline there, science confirms that redheads have genetic superpowers.
Yeah, it was very, very clickbaity. Proof that you should always think before you click that share button. Thank you so much for playing. I'll hop off now. Just a reminder to everyone to submit your questions and we'll get to them as soon as we can. Thank you.
Didn't do too badly. 3 out of 4 or something like that. Didn't embarrass ourselves.
Yeah, well, I think that it's very interesting because the headlines that come up in these sort of news reports sometimes are very hard to interpret in what's in science, for sure. Yeah. So anyway, David, that was a really nice sort of overview of where pain research is at today. And there's obviously, I think there's going to be a lot of questions coming in over the next little while. But I wanted to dig a little bit more. I know that you talk a lot about natural products, and indeed some of your earlier research is sort of, you worked with natural products. That's what got you into your research today. How much more is there out there? Is it an endless universe of natural products that we should be harvesting and exploring? Or in a world where we know the chemistry these days, can we develop more libraries of our own products?
I mean, we can certainly, synthetic chemistry is certainly a very powerful approach to pharmacology. There's no doubt about it. But I think the natural, my view, which is biased, would be to say that the natural world is still largely untapped. And I think plants and animals can make things that humans in a lab that are beyond some of the capabilities of humans to make. The beauty of natural products, and I should say that when you think about analgesic agents that we use today, the 2 main arms of analgesic therapy, so as I mentioned, opiates and non-steroidal anti-inflammatories, those derive from natural products. So of course, opiates are derived from morphine, from human experience. And NSAIDs as well, because it's willow bark that gave us, salicylic acid, which is a precursor to aspirin. So the natural world's been very good in terms of developing at least those 2 pain medicines. I think we have to explore all of these things, and I think now that there's more structural biology come into play, that is, we see what these molecules look like, we can understand how better to develop drugs. I think synthetic drug design and screening is now also extremely potent, but there's still so much out there in the environment that I think there are now efforts to go back in. There are a number of NIH institutes that are interested in mining natural products because they have turned out to be so effective for many of the drugs we take are derived from natural plant or animal products. I think some of the limitations are obtaining these things and connecting to the human experience that tells us where to look, and speaking to people from other cultures who maybe have thousands of years of experience with plants and animals in the wild that they've used, and really trying to get a more comprehensive overview of that experience and combine it with modern chemistry methods. So I think there's still a lot out there to discover. It's hard work doing natural product chemistry, and it's hard work screening it. So it's very expensive, and that's why it's kind of fallen off. But I think it's still could be a very fruitful avenue.
Yeah, you sort of mentioned it today, but I've seen it in some of your other talks and presentations. Mm-hmm. Anthropology as being an important part of this. You have to connect with cultures to really understand what products what they're mining from nature that is giving them pain relief or whatever.
Yeah, absolutely. Otherwise it's just a random search.
Yeah,
and given the number of things that one could look at, and every plant and every animal, every venom from a spider or a scorpion or whatever has thousands and thousands of compounds in it, and so that searching process could be limitless unless you have something to direct your gaze in a certain area.
So it does lead into sort of the issue around appropriation and working with native populations, and not. So that gets into a lot of ethical.
Yeah, I think there's definitely much more awareness of that now, on all sides. I think countries that have valuable resources are much more savvy about protecting them, and countries that want to go in and partner with scientists with their foreign collaborators are much more sensitive to doing that in a way that respects their provenance over these things, but also respects all the knowledge that they have to really make it more of a partnership. And I think especially younger people in this area who are prospecting for natural products are really going into it with that attitude, which I think is fantastic.
Yeah, that's really, really important. We do have some questions coming in, so maybe I should let go with a couple of questions, and then I'm going to come back with some more broad questions. Has your research touched on somatic pain syndromes caused by stress?
It hasn't so far, but we're starting to get interested in this area because one of the areas that we are interested in related to disease over the last 3 or 4 years is to understand visceral pain, which really in the context of what we're studying means gut pain, and pain that's associated with things like irritable bowel syndrome or inflammatory bowel disease. And I think stress plays a role in everything, but particularly in those kinds of systems, there's a big component of stress response. And there's also, a well-known interaction with neural stress systems, namely sympathetic nervous system. And so, we and others are trying to understand how input of sympathetic neurons to the gut can interact with cells in the gut and with sensory nerve fibers in the gut to regulate gut pain under different types of stressful conditions. So yes, we're interested in that. We haven't done much yet in that area, but that's a very important area of study.
The next one is asking you to comment on a specific approved migraine drug, frenesemel, whatever it is.
Frenesemel. So I should say, first off, I'm not a clinician, so I can't give you any clinical advice.
No, absolutely.
I'm just a basic scientist, as I say.
Can you comment on the sort of science of it?
I'm not familiar with the drug name, but I think most of those new drugs are drugs. Some of them are antibodies that minimize the effect of a transmitter that we just mentioned before, called CGRP. CGRP is released by nerve fibers that are involved in sensing pain, or at least a subset of nerve fibers involved in sensing pain. and they are also involved in in vasodilatory responses in response to activation of nerve fibers. So this is very important, presumably in migraine situation where nerve fibers that are associated with the vasculature are believed to be involved in initiating migraine pain. And so these new therapies target this peptide transmitter called CGRP as a way of diminishing migraine pain. And at least from what I understand from going to meetings and reading the newspaper, et cetera, these antibodies, they don't work for everybody who has migraine pain, but they can be very effective for people who have really severe remitting migraine episodes. So I think, again, this is really a great example of understanding the neurochemistry and the basic mechanisms that are associated with pain. People have studied, numerous labs have studied migraine pain for many years. And I think there was just a European Neuroscience Prize that was given out for this, and really trying to understand what specific types of sensory nerve fibers are involved in migraine pain, some of the issues that I brought up briefly in the presentation, and what are their molecular attributes, what kinds of chemical transmitters they produce, and what are the main players. And it's that kind of rational approach that's really led to the idea that antibodies against CGRP could be useful for preventing migraine pain. So I think that's a great example of a sort of a rational-based drug design in the pain area.
So are there other kinds of pain situations where the antibody approach could work? Because you talk mostly about sort of, small molecules.
One is antibodies that have been developed to something called nerve growth factor, which is also a protein that's important in early development for the development and trajectory targeting of nerve fibers involved in pain sensation. But later on in adults, it's also produced and can lead to can heighten pain sensitivity by sort of by hypersensitizing the nerve fiber. And actually, we've worked on some mechanisms by which it's been, nerve growth factor enhances sensitivity of some of the molecules that we've been working on. And because nerve growth factor and other such factors are produced at relatively high levels when there's injury and inflammation, again, people have targeted different components of this so-called inflammatory soup as a way of diminishing inflammatory pain. And nerve growth factor is one of them. And there are antibodies now to nerve growth factor that are in various stages of clinical trials for treating things like osteoarthritic pain. And there have also been some side effects associated with those antibodies, and then sort of retooling of those treatments to sort of deal with some of those problems. But I think some of the clinical trials have actually been quite promising. And in rheumatoid arthritis, there's an antibody against something called TNF alpha. That's also another component of the inflammatory milieu that you find in arthritic joints, and that's been a very successful approach as well.
And yes, I know that's a very successful approach. I hadn't really thought of it as being addressing the pain issue, but you think that
true. Well, yeah, it's true. It addresses more of the inflammation issue, but
inflammation is associated with pain. So I guess
and in rheumatoid and osteoarthritis, the thing that gets people most is the pain associated with it. Yeah. So if you can diminish the pain, even if you don't necessarily ameliorate the some of the other tissue damaging consequences, you've done patients a lot of good.
Absolutely. Yeah. Okay, back to the questions here.
Right.
Can you comment on the sort of ethical issues regarding objectively measuring pain and how it may affect pain patients' feelings of being believed? So I guess the argument being, I really am in pain, and if it doesn't show up on the image.
Yeah. Well, I think an objective, from my clinically naive point of view, not seeing patients, I would say that, or at least talking to friends of mine who are pain clinicians, I think that an objective measure of pain can only be useful because too often what happens is that this subjective measure of pain, it leads to patients not being believed. So unless, let's say you have an injury, somebody, you're playing soccer and you did something to your back and then everything's fine and you keep going and saying, but I still have pain. Oftentimes people are not believed. And because there's no sequelae or physical evidence of tissue damage. And so I think that's the worst situation, where you really are in pain and you're not believed. If on the other hand, there were a more objective measure that would pick up an index of pain, even when it can't be observed because there's no physical injury, I think that would be really beneficial to patients. Because I think right now the problem is that it's more often that people are not believed because there's no sign of injury. But if there was some biomarker or some neural signal that would say, yes, this pathway is hyperactivated, even though the sort of peripheral evidence for tissue injury has resolved, I think that would only be beneficial to patients in terms of giving more of a voice to what they're saying really is pain, and to leading to a way to not only for the physician, but for the patient to actually monitor their own progress.
And how close are we to having good objective measures of?
I would say we're not super close.
I
think it's a very complicated goal. I think this whole idea, the NIH and the National Institute of Neurological Disorders and Stroke is really trying to put some emphasis on finding biomarkers, but I think we don't yet really know exactly what those biomarkers might be, aside from certain, transmitters, et cetera. So I think, that's a push to try and really just do the basic research of trying to see if there are molecules whose expression goes up and down, et cetera, that are associated with certain types of pain syndrome. So I'd still say we're in the early stages of that. And for imaging, I'd say, the same thing is true. I mean, there's certainly Catherine Bushnell and other people have, done some really beautiful work over the years to look at areas of the brain that are activated when there's, activation of peripheral areas. And I think that imaging technology is moving along so quickly in all realms of biology and medicine that I would say that, I'm hopeful that those areas will really blossom into something that can not only be used to more objectively measure pain, but to also be coupled to, at some point, biofeedback devices and things like that. But, it's at the very early stages, I would say.
Okay, the next question is about marijuana or THC pain relief.
Yeah, I don't know. This is an area that, I've talked to a number of people about this. I think this is an area where the clinical assessments are also still in the early stages, and whether there's really enough sort of controlled trials to know whether cannabinoids are truly beneficial for controlling pain in sort of an objective way. Some people, this is where I think the subjectivity of pain control and the variability from person to person is both confounding but important to look at as well. And I would say that the same thing holds true for treatments like acupuncture, et cetera. Some people find them to be incredibly beneficial, and other people not.
Down the bottom, actually, somebody's asking about that.
Oh, there we go. Yeah. So I'd say for THC and marijuana, I think many are awaiting more sort of rigorous controlled studies on this. For things like, I have friends who swear by acupuncture, and there's a whole part of the NIH, the NCCIH, that's interested in looking at non-traditional approaches to deal with pain. And I think those things are very important and interesting to look at, as long as they're done in as well controlled a way as possible. Again, here is where having more objective measures would be really helpful. Yeah,
it does sound like that is a very important goal. The next I'm missing, so the question comes up, which you sort of addressed, but somebody's asking much more specifically whether there's a better pain med in development to overcome the negatives of fentanyl.
Fentanyl, yeah. So, opiates are amazing drugs. People take an opiate in their pain. They are amazing analgesics, but we all know what the downsides are in terms of either long-term addiction and tolerance, or short-term overdose issues like respiratory depression. There's a lot of effort trying to still not not give up on opiate systems as important targets for treating pain because they're so well validated in terms of basic studies. But in finding drugs, this is kind of what I was alluding to in this one slide, where there's a push to find what people call bias agonists, which are drugs that work on opiate receptors, the same receptors, say mu-opioid receptors that morphine works on, but to activate the receptor in a way that will enhance the signals that lead to analgesia at the cost of, or rather than also activating signaling pathways that lead to things like respiratory depression or constipation. That's still sort of a controversial area and an area in flux, whether that can be achieved, whether you can, and this all depends upon whether the receptors for these opiates engage in different conformational states when they are activated by different drugs, and whether this has a downstream outcome in terms of how they signal. That's kind of the hope, but I would say that that's still also in its early stages. But looking for drugs that act at known validated targets, but work on them in ways that are different than existing drugs is kind of a pathway that people are mining and looking at from both computational ways and real physiological ways. But so far, I'd say there's no drug in the wings that really takes advantage of that kind of a mechanism.
So can I ask a little bit, sort of follow up on that in terms of, so we talk about people doing this. Mm-hmm. Obviously, the big drug companies, we know several of them really, put their money into opioids and led to the opioid crisis.
Yeah.
Are we looking at small startups trying to go in different ways, or are we looking at big pharma actually engaging in trying to replace the things that have been?
Yeah, that's a good question. I think from what I've seen over the years, and again, I don't have close ties to the pharmaceutical industry, so I'm not an expert, but I think pain has always is a difficult space for big pharma. And many big pharma houses have dropped or diminished their pain programs over the years, despite the fact that we now have a lot more information. And I think that's in part because, there are other areas that are competing for attention, like Alzheimer's and cancer. But also this comes back to, how do you assess pain in clinic, new analgesics in clinical trials? And, I think one of the stated goals has always been over the years that new analgesics have to do at least as well, if not better, than morphine or an opiate. And I think in light of the opioid epidemic, that realization is changing. And I think maybe that's a healthy thing, because I think it's an artificial bar. And the other problem is that I think we have to think about other types of models in clinical trials to look at. Not just things like molar extraction or bunionectomy and things like that, because there may be some drugs that we've looked at and rejected over the years that might be very good, for example, for treating, let's say, fibromyalgia someone's written about, or treating migraine or visceral pain, that didn't score well in some of the very standard models that people have used. So, in pharma, I think, I'm sure people are thinking about all these things, how to reassess different types of clinical models, how to go back in some well-validated targets, but hopefully, I'm hoping that all this discovery of new targets, like some of the things we've been working on or many other labs, will now be picked up by both small and large pharmaceutical firms. I think it's a great time to come in if people are interested in doing a startup that's focused on pain medicine. I think there's such tremendous room. It's a hard area to work in, but I think the outcomes could be fantastic. And I think that it's a space that some large pharma houses have given up on in lieu of other targets that are also important, but perhaps more approachable from a business point of view or other points of view.
Yeah. Okay, back to the list. I think
okay.
You mentioned the next one is about fibromyalgia and neuropathic pain.
Yeah.
And so comments on that, suggesting that somebody wants a review paper for this. But.
Yeah, I don't have a review paper off the top of my head, but there is a lot of work on things like fibromyalgia, trigeminal neuralgia, other types of neuropathic pain. We study mostly inflammatory pain, which is at least at some level different. And, I've always focused on that because of the molecules that we've identified are more associated with those types of pains. And I find that sort of pain to be, at least from my perspective, as a biochemist and molecular biologist, a little bit easier approach because we know some of the factors that are likely involved in those types of pain. Neuropathic pain, I think, is still a more difficult space, and there are some good models that people use for that, to study that. And I think now with some, more enhanced gene profiling methods, et cetera, we're beginning to, a lot of people looking for molecules whose expression is affected in different types of neuropathic pain, such as, for example, diabetic pain, pain that results from from taking chemotherapeutics, and other types of neural injury pain or cancer pain. But it's still an area that needs a lot of work. So we don't really understand that there is some understanding of some ion channels and other molecules that are disrupted in different types of neuropathic pain. But I think we're still, at the very beginnings of trying to understand that.
So back to the placebo effect.
Placebo effect. Yeah. Well, there's a very famous paper from UCSF years ago from Howard Fields and colleagues that showed that the placebo effect is diminished when people take naloxone, which is a drug that blocks opiate receptors. So the idea has always been that placebo effect is a real effect. And what that means is that one is harnessing your own endogenous pain control systems, which is natural endorphins and enkephalins and things like that to control pain, which is a very fascinating concept and something that, you think could be exploited more. So I think the placebo effect is believed to be a real physiologic thing. And there is great interest in trying to understand the pathways that underlie the placebo effect, because again, that's something that one would like to harness. I mean, this is in part maybe, related to the use of neurostimulators and things like that to activate pathways like descending opioid pathways that are normally activated when one experiences the placebo effect or mounts a normal analgesic effect, at least to, intense acute pain experience through, for example, mounting an endogenous opioid peptide response. And so I think the more we understand about the placebo effect, the more we'll understand about endogenous systems that control pain.
A couple of quick, more specific ones. This is, I don't know if you know this one. Are you using pain trace?
We're not, but I'll look it up and see what that is. Yeah,
absolutely. And substance P inhibitors.
Substance P inhibitors. So substance P is a compound that's released, or a peptide that's also released from sensory neurons together with CGRP. And substance P elicits this phenomenon called vascular leakage, which again is another action on the circulatory system that allows circulating cells like mast cells and basophils and other cells to enter the area and contribute to this process of neurogenic inflammation, and has a double-edged sword. It helps to resolve damage and infection in the area, but it can also lead to inflammatory pain. I don't know of any clinical trials. I don't know of I'm sure there's some out there in terms of substance P inhibitors. I know that early on, mouse studies in which substance P or substance P receptor were inactivated did not show a very large effect, in pain models. But that was done a long time ago, and there are now better ways to do those experiments. And to be honest, I don't know if people have gone back to look at the role of substance P in particular types of pain syndromes. But all of the transmitters that are released by the sensory nerve terminal in response to damage, I think, are all potentially interesting targets for pain. And I think this is another example where it's probably worth going back and looking at some of these molecules like substance P in different types of models of pain sensitivity, because my guess is that if they've been looked at, they've only been looked at in some very specific models that may not be the right setting for picking up a noticeable effect of inhibiting these pathways. So,
excellent. Well, we've covered most of the specific questions, and we have a few minutes left.
mm-hmm.
I did promise you that I was going to sort of query you a bit about your career and how you got where you were today. And I know that when we talked with the Gairdner Award, you said very much that you were driven by curiosity, that you were curious about the world, you were curious about the natural world, and that led you into these specific pathways. So tell me about your early curiosities.
Well, I don't have a story like one of these kids who says, well, I was always fascinated by little bugs in the backyard. I think, I wouldn't say I was indifferent to that, but I was interested in a bunch of different things. I think my curiosity about science really got piqued when I was in high school. And it really got piqued when I took a great physics course. And the teacher really drew me into, using my imagination and some developing quantitative skills to try and really see how the natural world can be described and explained through reason and curiosity and to some degree mathematics and things like this. And that really sort of focused my attention on science, I think. And then when I went to college, I knew I would never be a good enough mathematician to really be a physicist, at least not a theoretical physicist. But when I got to college, I became interested in biology, and I thought that I would be a clinician, go to medical school. And then I did something important, which was I decided, I was bored going to classes all day. I wanted to see what it was like to work in a laboratory. So I signed up to work in a lab as an undergraduate volunteer, and that's really where I think my head really got directed towards basic research. And I think it was for two reasons. One is I really found that I liked experimental science because I really enjoyed working at the bench. It was like having a hobby that was also your job. And, it was a great combination of thinking, logical thinking, and also, dreaming about stuff and having these models and ideas, but then also going in and doing something that with your hands where you could really sort of translate those ideas to something physical. And the other thing that I always thought that I remember one day finishing an experiment and putting my experiments into this, machine scintillation counter where you can read like radioactivity coming out, and a strip of paper is coming out of it, and it has all these numbers on it. And then I take it back to my desk and I'm sort of plotting this. And then I'm thinking, this is really incredible. I've set up these little biochemical reactions in a tube. I really can't see what's going on in the tube. I can only kind of read these numbers out and then, using your imagination, kind of extrapolate to what you think physically is going on in this little test tube where you can never actually see anything. And this idea that you could kind of design an experiment, see the numbers come out, and then put it into sort of a working, model and a framework of what you think is happening, and then be able to then go to the next step and test that model, to me just seemed like a beautiful thing. And so I think it was really actually doing science that convinced me how beautiful it was. And that's really, I think, when my curiosity got piqued, because I started becoming interested in more and more things as I understood the kind of questions you could ask and how you could actually solve some of these problems. So, and today I even tell people in my lab, most of what you try during the day or during the week or even during the month is failure, right? It doesn't work. And, it can be very trying sometimes. But, at the end of the day, when you go home, sometimes you just say, I don't understand what's going on yet, but my experiment today was so beautiful. I ran the most beautiful gel, or I did the most, whatever it is you're doing in terms of your techniques. And that sort of physical aspect of doing that and having the beauty of designing experiments and, is really, I think, what got me going. I think in part it was also a big respite from sitting in classes all day. And it really gave me, it really gave me some space to breathe and to kind of see science at the real, life level.
But you really were, and you are, basic science. You want to understand how things work. But as you've just demonstrated to us tonight, understanding how things work is what you need to know in order to be able to develop new treatments in a more rational way. And I think your talk tonight really emphasized the importance of that. And certainly, even to me, I hadn't really thought about quite how pain, just pain alone, is so variable and so different in different parts of the body with different sensory inputs. It's a challenging area to be working in, for sure.
It is a challenging area.
You're still hitting it with hammers instead of very fine approaches. Right?
It is a challenging area, and you can really see that when you speak to clinicians, pain clinicians who see patients, because so many times they're at a loss to identify what the problem is, or what the initiating injury was, or how to treat the patient, or the patient has gone through multiple different types of treatment scenarios and it hasn't helped. And so you can see the complexity of it there at the human level. It also is complex at the biological level, but I think all of physiology is complex at the biological level. And what we do is, as you well know better than almost anybody, what you try and do is to break it down to some basic scientific question that you can address, and then bootstrap from there as you get clues and work your way up. But it takes time.
Yeah. So, I mean, really, it's been a wonderful evening. It's great to spend some time with you. I hope the audience has enjoyed spending time with David and hearing about his research and where pain research is going today. So thank you very much indeed. And this is great from the point of view of Gairdner. Every year we partner with RCI Science to bring a Gairdner laureate to the stage, usually in person, and hopefully we'll be back doing it in person next year.
We'll do that in the future.
It's also great to do it in this webinar format as well. So thanks, everybody.
All right. Thank you very much. It's a pleasure. Nice to see you all. Nice to see you, Janet.
Bye.
Stay well.
A huge thank you to the Gairdner Foundation, Dr. Osman and Dr. Julius for joining us this evening and sharing their insights into the world of pain research and pain management. I hope you enjoyed the discussion as much as I did. If you can, and if you're able, please do consider supporting RCI Science's free programming by making a donation. All donations, however big, are hugely appreciated to us. And you can do that easily by visiting our website, rciscience.ca/donate, and there'll be a link in the chat. Please do share your feedback with us and let us know what you'd like to see from us in the future. We will be planning our schedule for heading into fall 2021. It's quite likely that we'll still be online, so we'd love to get a sense of what you'd like to see us tackle next. And again, a link to the feedback survey will be available in the chat, and email to those of you who did register. Science Odyssey, which is a festival of science taking place across Canada, continues next week, and with that, so too does our programming. On Tuesday, May 11th, we joined the University of Ottawa's Institute for Science, Society and Policy to take a look at the impacts of embracing AI and machine learning for creativity. And that starts at 5 p.m. Eastern. And then on Thursday, May 13th, we'll be joining the Queer Women of Esports Twitch stream, which is pretty new for us, for a look at how video games can be used to support scientific research through citizen science. Specifically, they're going to be playing Borderlands Science, which is a mini arcade game found within a really popular video game called Borderlands 3. Believe it or not, this is a mini puzzle game that organizes data so scientists can map bacterial DNA in the human intestinal tract, the digestive system. And we'll also be joined by the scientists and the game designers behind Borderlands Science. So that should be a really, really fun and exciting discussion. As always, details of all our upcoming events can be found on our website, rciscience.ca/what's-on. There'll be, again, a link in the chat and a link on screen as well. But with that, I will let you get on with your lives. I thank you so much again for joining us today. I hope to welcome you to an RCI Science event in the near future. Take care and all the best.